on early eukaryote from Torridon. And then we'll follow up with Bo Jones, who's going to talk about the Cretaceous-Paleogene boundary in Antarctica. And then we'll go all the way back to the Archean, where Steve Hollis will talk about mineralization in the Australian Craton. So 3 very diverse talks. And yeah, I just like to say the usual fire escapes are there. Please don't close the door of the one on the left. It's open to allow us access in as well as out. But if there was a fire alarm, you can go out there or here. But without further ado, I will introduce Edwin Dzul to talk about the early eukaryotes from Torridon. Go ahead, Ed.
Thank you very much. Hi, everyone, and thank you for allowing me to give this talk to you. And this is actually like a different version of a talk that I gave before, but it's going to be quite interesting for me to like put together all the information that I collected from a field trip that it was actually granted by the EGS. So that's why I'm very happy to be here as well. So I appreciated all your help. So yeah, my talk is about non-marine early eukaryotes, and I'm just going to talk to you about a little bit of this work that has been now published actually this year. And it's my first my first author paper, and I'm just quite happy about it because it's been like cooking itself for like 2 years. So yeah, so this work is a systematic description of microfossils that are from this magnificent assemblage that is the Torridon Group, specifically from the Diabaig Formation that is dated about 1 billion years old. And this is right here in Scotland in the northwest, just in front of the Isle of Skye, you know, but in mainland. So it's in that area. And before diving fully into these like magnificent microfossils from the Torridon, I'm just going to give you a brief introduction of something that we all know. Life's living forms that we can find here on Earth. And we know that we have 2 different types of cells that we can find here that are making up the life. And those are going to be prokaryotic cells and eukaryotic cells. So prokaryotes are actually found in 2 different domains of life, which are going to be bacteria and archaea. For example, in the top right, we have cyanobacteria, and at the bottom, we got an archaea called Allobacterium. So these are unicellular, mostly microscopic organisms that they don't have organelles or a nucleus, compared to the eukaryotic cells, which are all the macroscopic animals that we know and we can see with our naked eyes. For example, us, or like bugs, bears, fungus as well. And all of these are coming from the last universal common ancestor, and later they develop into different domains of life: bacteria, archaea, and eukaryota. So the main goal of my research in general, and actually from this paper, is trying to understand when the eukaryotes evolved and how. And this is actually one of the most problematic and major events that happen in the evolution of the history of life on Earth. So quite complicated. For this, and for us to try to understand how these eukaryotes evolved, we are looking at the fossil record, something that it's called organic ball microfossils. These are remains not just of eukaryotes, but also prokaryotes, and they are going to be preserved as compressed carbonaceous vesicles, also like empty sheets or empty blobs, for example. We got some magnificent examples here as well of both, all of these are eukaryotes, and this is a prokaryote, just because the main point in order to tell apart between prokaryotic organic wall microfossil versus a eukaryotic is the morphology. Sometimes morphology alone is not all for us to identify them, but that's the first step that we take. And if we have, for example, a very complicated morphology, we are towards labeling it as a eukaryotic microfossil. And these were called traditionally architracts. Actually, in some books as well, they still calling them like that. But morphologically speaking, they are going to be more complicated than prokaryotes. And the fossil record of the early eukaryotes is quite scarce, but there's research about it. And we got, for example, that the oldest red algae has been discovered, and also the oldest green algae, oldest fungi. But all of these are just like compiling the information for us to like build up this history of the evolution of early eukaryotes. So that's why it's important for us to take a look into these fossils, because not just because they are important for the evolution of all life form on the planet, but also because they are going to tell us when the first crown group eukaryotes evolved. And that's quite interesting, because in that way we are going to know when the first, for example, brown algae appear, which we don't know so far, or how the filamentous fungus started to evolve. All these questions that we are still trying to understand. The fossil record is going to help us in order to understand that. And how are we going to investigate where or what it could be, tell about the eukaryotes? Well, we are taking a look on the fossil record, particularly to the Proterozoic, and scientists are using different approaches, not just the fossil record, but also like molecular phylogenetics, also organic biomarkers, proxy, redox as well. In my case, I'm using experimental taphonomy, also vibrational spectroscopy techniques. But the main point is first looking at the fossil record. That's the idea that I want to, to bring to the table. And that lead us to the importance of studying these microbial fossils, here specifically in the Torridon. There are three main reasons for that. First of all, the age of this assemblage is very special, because it's dated between the Mesoproterozoic-Neoproterozoic, which is at a time when it is believed to be the radiation and the evolution of the first crown group eukaryotes. And also, the second point is the exceptional preservation that we are going to see here in the Torridon group, specifically in the phosphorites or phosphatic bands and nodules that we are going to have in here, alongside of these like shales that we are going to find here in this environment. And all the microbial details of these organic wall microfossils, well, when they were alive, they got exceptionally preserved. So you can see them under the microscope, and you can see inside of these cells, for example, and also the filaments. So it's quite amazing. It's a very cool site to investigate. And the last point is the environment, because this is an inland aquatic environment. It's still debated, and I'm going to talk a little bit more about this later, like briefly, but it's still debated if this was a fully freshwater environment or an estuarine environment. There are some like recent publications debating this, but nevertheless, it's a very specific environment, inland, and this is quite rare to find in the fossil record. So what we've done in this paper was investigating the rock samples, making some thin sections, and building up in previous work done in this site. We are describing 11 morphotaxa between old described fossils and newly described as well. And particularly, there is one that is a new specimen, a new species, a new genus, sorry, that is called Minimarmilla multicatenaria, which is the one that is here. It's a beautiful fossil. And it's enigmatic because we don't know if this was from algae or a cyanobacteria or something else. So we still don't know, because we don't have the specific features of any of these two groups in order to tell apart. But nevertheless, it's a very cool fossil. And the main point of the paper was to do this systematic description for the first time, because even though there is some very excellent previous work done on the site, nobody have done before systematic description. And we believe that doing this is particularly important for us to have a catalog of fossils. So you can go to the assemblage and look what specific ecology you are going to have there, what kind of organisms were living there. So all of this is like a starting point for future work to be built up on this. So I think that's why it's important to have systematic descriptions. And talking about a little bit of the Torridon Group, and specifically from the Diabaig Formation, that is the site from where we took the samples. This was a Meso-Neoproterozoic dated place around 1 billion years old. And it's a non-marine paleoenvironment, well, it's still debated, as I mentioned before. And the Diabaig Formation is actually the lowermost formation, of all the Torridon Group. So now just talking about a little bit of the field trips that we've done in here. Well, actually, I participated in the number 2, but the number 1 was the main point of it was the starting point, for this whole research, because my supervisors took some samples from this place, and they found out that it was a good possibility of continue investigating this place in order to find more fossils. So that's why we applied for the grant for the EGS, and luckily we got it, and we were able to go again on October 2024 last year. So the results so far are on the work. So hopefully next year we are going to, I could come back and like update you about this. But I just wanted to show you like the amazing phosphatic nodules and bands that we can find in here. And also we got some reticulated cracks, and these are indication of some high energy environment that it was occurring there that is beautifully preserved, and among other things. But basically, I just wanted to show you what we've done in the field trip, yeah, based on that. And these are just like beautiful images of this place, the Torridon Loch and Loch Mary and Loch Shieldaig. Sorry, I have problems pronouncing the names, but beautiful place. I highly recommend it to go for a weekend away. But coming back to if this was a lacustrine or an estuarine environment back in the day, 1 billion years ago, is still debated. We got some research stating that this loch was formed by rivers and streams that came from the mountains and like unfilled this whole place. There is also other research stating that channel belts were the main point for this place to become a loch. But there is this recent paper that built up in previous work as well, stating that by checking the salinity indicators of the rocks of this place, is telling a different story, that it could be an estuarine environment, just based on the fact that you could have like a seasonal input of marine water into this like isolated loch. So that's still debated as well. So we don't know. And actually, it's not the scope of the paper, but we wanted to show this because it's important to know where our ancient organisms were living. So anyway, the Torridon is a rare archive of this Proterozoic non-marine environment, which is quite rare in the fossil record. And just like stating quickly how the microfossils got preserved in this place, this is still under research as well, because it's not really fully understood how the phosphate come together and preserve beautifully these microbial organisms. But this hypothesis stated by Leah Battinson in her dissertation was stating 3 steps where basically you got the microbial organisms in the water column. They were clay minerals around them, and these clay minerals created this cast around them based on all the minerals that we're getting on the cell walls. And after that, the phosphate precipitate around them, and that prevent them the further decay. And after that, the preservation allowed them to become fossils. So that's quite interesting for this specific place, because you can raise questions about like where exactly the phosphate was coming on, and if, apparently there was a lot of phosphate there. So yeah, there's still like key things that they need to be addressed. But the main point is that the early formation of the clay minerals allowed this rapid precipitation of the phosphate around them and made the preservation to occur, which is quite interesting. Is beautiful. And these are some of the examples of these microbial microfossils. And we got Leiospheridia, which is just like a vesicle, but you can see inside and also some cracks as well in here. And we believe that this was an existment, like a hole where the actual organism, when it was alive, just went out, and it got preserved just this like a structure in there. And we got as well this massive colony of Siphonophycus, which is a microbial mud, more likely from cyanobacteria-like organisms. So this is quite cool as well, and it's like massive in our samples. But yeah, we got a high diversity of these microbial fossils here in Diabaig Formation. In the paper, we are stating that we had two unambiguous eukaryotes, two possible eukaryotes, and this new filament-forming taxon, Minimarmilla multicatenaria. And we got in here as well all these taxa that we described, and two eukaryotes for sure, which are going to be Bicellum brassieri and Germinosphera bispinosa. I'm going to show them a little bit later on. And two prokaryotes, Eukaryotic and Siphonophycus as well. And all of these in green, we don't know exactly what could have been, but we got some clues to say that this guy in here called Pteropsimorpha, and also the unnamed species B, they could have been eukaryotes just based in the fact of the complex morphology they are showing. And we compared this as well with the previous reports of some assemblages of the same time. And even though the Torridon is not really massively outstanding compared to these other assemblages, it is the oldest known aquatic microfossil assemblage. So that's quite interesting to know. And it's just a little bit behind of other assemblage that is the Munjabi and also the Atari Elementary group. But this is just for us to see that the Torridon group is like a very special place to go and try to find and identify these organic wall microfossils. And now I'm just going to show you some of the images of the microfossils that we described. Not all of them, but just like the key ones. Minimarmilla multicatenaria, for example. And the name, it means a small bracelet just because it looks like this, like a rectangle-like beads that you can find in some like hand bracelets. So I think it was something cool to work around on. And we don't know exactly if this was from algal or bacterial origin, because we don't have specific morphologies that are going to guide us towards one of these 2 points. But we got some clues, for example, in here in these two images, 15 and 16. These look a lot like these reproductive structures that you could find in red algae, but this is sadly the only example that we had. So we need to hopefully find more specimens in order to build up more this story. And these are more like beautiful images about it. You can really see here like how the cells got preserved in what it was at some point a sheath around them. So that's quite cool. But also like lots of them that are like all compiled and following the same direction. So this was definitely something that if this was just a cyanobacterial community, is quite incredible how they are just following that pattern, right? But anyway, there's debate about it. And another one, Eukaryotic lacustrina. This is a coccoidal colony-forming organism, and this is found just in the Torridon, but also in another place that is called the Non-Suc Formation. And this was first described by Stroder and Wellman in 2016. And this raised a question for us when we were doing the paper. it resembles a lot of types of coccoidal colony-forming organisms. So what we did in order to just to be sure that this was exactly this fossil that they were mentioning in the paper, we just make a comparison using the length and the width of this organism. And by doing that, for example, the Eukaryotic lacustrina from our study compared with the original description, if you take a look of the image, when now the cells are highlighted, so those are ours from our study, we compare them with the original description of Eukaryotic lacustrina from the Torridon as well. And we can see that the size, it's matching. And also we compare them with Eukaryotic lacustrina from two other places. And, and they are really a little bit more like a constrained specifically with the original description of Eukaryotic lacustrina. So just based on this morphology, we could tell that, well, it is what they were saying that it was. So, so we just roll with that, and we continue calling this a taxon like that. And now, just like some final images of some of the fossils as well that we can find there. The two eukaryotes that we know that they are for sure, just because, again, of the morphology that we are looking at. One cool one is Bicellum brachieri, which is, it's believed to be an early stage of some holozoan. So it could be an embryo, for example, and the main point of for us to tell in this, based as well on the original description by Stroder and Wellman, is that it's composed by two different types of cells. We have in here, for example, inner cells and outer layer cells that they are forming this like massive structure. It's like a ball made up of different cells. And lastly, the Germinosphaera bispinosa. This is, it could have been, and is still debated as well, if this could have been from a fungal origin, but we cannot tell just based on this. But what we can tell is that it was complex enough to be labeled as a eukaryote. And we got other two fossils as well, new ones, unnamed species A and B. This is like a beautiful preserved ball of filaments that we believe that it could have been from cyanobacteria. And this unnamed species B, which is sad, but that this is literally the only good example, the figures 4 and 5 that we have, with a vesicle in here attached, that it could have been from fungal origin as well. But we don't have more examples in order to tell apart this story. So yeah, it's debated. And the conclusions now, sorry, I think it's like a long time for me talking, but yeah, the conclusions are that in this work, we describe 11 morphospecies of these organic wall microfossils that we can find here in the 1 billion years old Torridon Group, specifically from the Diabaig Formation. And this is a work that is willing to add up more information about the Torridon research that has been previously done. And we describe in here 3 new fossils, Minimarmilla multicatenaria and the unnamed species A and B. And that would be all for me. A special thanks to the co-authors of this paper, of course, my supervisors and my colleagues as well. So thank you very much.